Fantastic. I think you did a really, really great job, and well done, everybody.
That's it. So thank you again.
Wait 1 second, please. So very well done. Good job. We really enjoy listening to you, your proposal, your reasoning. A very big thank to Russ for the work that he has done to prepare everything, and to Marco for organizing things. So now the program is supposed to proceed in 1 hour time, more or less, with the assembly of the young chemists, the members of the young, the young members of the Italian division of medicinal chemistry. This is for Italians. But before the assembly, we have the very nice occasion to deliver the award for the so-called ESMEC Alumni Award, which is a small recognition that we give to scientists that have been part of the school in previous years, and that have exploited, hopefully, what the school and the PhD program has given them for their future career. So tonight we have the recipient of the award could not join us here in Urbino because she's in Singapore, so it's a bit far. So she is present here remotely. And it's my pleasure to give the floor to the president of the Italian division of medicinal chemistry of the Italian Chemical Society, Professor Stefano Alcaro, that will present and deliver the award to the winner. Please, Stefano.
Okay, so first of all, I would like to thank the director, Gabriele Costantino, for this great edition, and all the staff that really is doing a very good job. I'm enjoying with you the presentations, the workshop, the lectures, and I strongly believe that for us, for the Medicinal Chemistry Division of the Italian Chemical Society, the school is really an important moment of formation, and it is really a pleasure for me. It is my first time here as president of the Medicinal Chemistry Division, saying that we try to support as much as possible the presence of you young investigators with the grants that some of you have been awarded, and now it is also my pleasure to go ahead with this special moment, because also the so-called Alumni Association, a group that is related to the ESMEC, in this edition has another award. And so I will introduce the recipient, but only after saying that we have, as you see in this slide, a couple of interesting options for you young investigators, because the registration for the national meeting that will be held in Parma from the 14th to the 17th of September is still open with the yearly registration that is active up to the middle of this month. So I recommend you to apply. But also for some of you that maybe can be interested in another option that was, by the way, a last-minute workshop that we decided to create at the end of a project related to the consortium MINFACT, that is the PAMVID, the international meeting in drug discovery against pandemic disease. I invite you also to consider this, and you will find in the two links that you see in this slide, the option to eventually, participate also with flash communication that I believe for some of you could be really interesting to give. I believe that also during the young group of the medicinal chemistry division that will be actually in the assembly that we have in half an hour, more or less, you will say more information about this, and also tomorrow I will just remind you about this option. But now it's my pleasure to go straight, since we are a little bit late, to introduce Professor Giorgia Pastorin, that I hope you can hear me. Giorgia,
I can hear you.
Okay.
Hi everyone.
Yeah, so first of all, we have this special guest that now is in another part, in another continent, because as much as I know, now you are in Singapore, right?
Yes.
Perfect, okay. And it is really a pleasure to me to introduce you because you, first of all, have been, you reached the degree in medicinal chemistry and also the PhD in pharmaceutical science at the University of Trieste. But also during your PhD, you have attended the ESMEC school. So that's why we have the pleasure to host you in this special session that is devoted to the alumni that obviously have been already participated the ESMEC school. I also want to mention some other information regarding your career, because you followed as a postdoc in Strasbourg in the group of Alberto Bianco, moving, as much as I understand from your CV, from the so-called drug discovery to drug delivery. And this is also an added value that it is now probably the reason why you are at the National University of Singapore, that is hosting you as full professor and dean of the Department of Pharmacy and Pharmaceutical Science. This is what I'm reading from your CV, and I congratulate you with this really great career. I also would like to mention that the scientific production of Giorgia is quite strong, with more than 160 publications and the H-index of 52. It also has been awarded by the Biodrome Award and the Bibleo Award. She's also within the top 2% scientists worldwide published by the Stanford University. So I believe that you really have a great curriculum, and it is a pleasure to introduce you, and also eventually to introduce yourself if you want to add more information for the young investigators that are here in the audience, and also with the title of your presentation that I believe now will be put in the screen, right? So, Giorgia, I congratulate you, and first of all, I would like to welcome and leave the floor to you, please.
Thank you very much, everyone. I'm really deeply honored to receive this ESMEC Alumni Award from the committee, in particular Professor Gabriele Costantino, the ESMEC director. And really, I would like to express my sincere gratitude, because I feel that being a part of the ESMEC community has greatly enriched my professional growth. And I thank you for continuing to inspire excellence in medicinal chemistry. A few notes about myself. I actually prepare a presentation. I hope you don't mind if I share my screen. Let me know if you see. Can you see the screen? I hope.
Yes, not now in full screen. You can Okay, now it's perfect. You can go
Yeah,
ahead.
done. Is perfect. Okay. As was mentioned, I go quite quickly because this was already covered. I got my master degree from the University of Trieste, and then a PhD in the same university. And I'm very grateful to have had a supervisor, Professor Giampiero Spalluto. Actually, this is the only photo that I could find of that time together. I was his first PhD student, and this is Paola, one of our first laureandas. So at that time, Prof. Spalluto was a young professor, very enthusiastic, and we had a very energetic addition of a very talented, at that time, student, but now is Professor Stefanie Federico, that I'm very grateful to have, that she was part of the team. And what I really find and I feel like sharing with you is that one particular aspect of that group is that was very collaborative in nature. And in fact, we collaborated with Professor Barbara Cacciari, Professor Stefano Moro, Professor Paolo Tecilla, Tatiana Daros, and many, many others from different universities. And you can see that this one actually is my first paper, not as a first author, I'm somewhere, here, but it's quite important to me because it includes the pyrazole-triazole pyrimidine that became a little bit the focus of my PhD. And around that time, I also attended the European School of Medicine and Chemistry in Urbino, and probably was around that time that I was very intrigued by the relationship between, professors, students, and it was a very vibrant type of interaction. And I decided that's probably what I want to do when I grow up. And therefore, that's why I basically started thinking about my academic career during that exact period. And as mentioned, then I went to the National Center for Research in Strasbourg under the supervision of Alberto Bianco, and at that time I switched to drug delivery. We had a very fruitful collaboration with Professor Maurizio Prato and Professor Costa Costarellos, and at that time I was working on carbon nanotubes. So I could see, for example, this carbon nanobottle where I included here drugs, I protected them, and I further sealed them with some fullerenes that acted as corks. And then since 2006, I've been at the National University of Singapore. Our labs, in fact, are in the last 2 floors of this building. It's quite a new building. But as you can see, quite a modern, vibrant university. And Singapore, per se, for those that are not very familiar with, is a very modern city. It is a country. It is a country 30 kilometers by 40 kilometers, so quite small. This is the circuit of the Formula One. So every September, if you're interested, that's where we have the Ferrari going around. And then is also there is quite a bit of greenery. There are many parks. Some of them are artificial, as you can imagine from this picture. And then we have orchids, because orchid is the flower, the national flower. But I didn't come to Singapore. As you can see, Singapore is called also Red Dot because in the world map, it's just a dot, nothing bigger than that. As I was mentioning, it didn't come to Singapore for the landscape, but rather for the university, because the National University of Singapore is very highly ranked. It's actually in the QS World Ranking 8 in the world, and it is the first in Asia. So we have about 30,000 undergraduate students and 20,000 postgraduate students. And the university is basically run by less than 3,000 full-time academic staff. This year, we are celebrating our 120th anniversary. So by Italian standards, this is a very young university, but by Singapore standard, is the oldest university. And this because Singapore is only 60 years old, 60. And these are just some of the numbers in terms of output of a very research-intensive and aggressive, in that sense, university. Now, Since last year, I have been heading the Department of Pharmacy and Pharmaceutical Sciences, and this department is within the Faculty of Science, and we are about 40 in terms of academic staff and 25 of support staff. And the majority of our students, as you can see from this number, are undergraduate students. And the reason for that is that we have two undergraduate program, Pharm Science and Bachelor of Pharmacy, but basically we are the only provider of pharmacy degree for the whole nation. And that's why we have the mandate to produce pharmacists for Singapore. And then we have also several postgraduate courses. We have two master programs, a master in clinical pharmacy and a master of pharmaceutical sciences and technologies, as well as PhD and PharmD degrees. And similarly to the university, if we look at pharmacy by discipline together with pharmacology, we are also first in Asia, and this year we jumped from position 14 and now we are number 8. Now, besides this, when I went to Singapore, I decided to keep a little bit both the medicinal chemistry as well as the drug delivery part. Now, in the case of the medicinal chemistry, I'm particularly focused on platinum-based drugs. And this for the simple reason that these drugs actually they were discovered in the 70s. There are 3 FDA-approved drugs, cisplatin, carboplatin, and oxaliplatin, and they are very, very potent. They are, chemically speaking, as you can see, quite simple. But as you can see here, they are a first-line treatment in many types of cancers, and this is a non-exhaustive type of list. So while they share some similarities in form of structure, they also share a mechanism of action. They form, in fact, they interact with the DNA. So the DNA in the nucleus is the biological target with which they form inter and intra strands that then will affect the replication of cancer cells that then will undergo apoptosis. However, if from the mechanism point of view, or from the structural point of view, they are similar. In terms of side effects, are actually quite distinct. Sorry, can you hear me?
Yes, yes,
okay.
Definitely.
Sorry, thank you. From the side effects are quite distinct. Just to give you an example, in case of ototoxicity, so the toxicity in the ears, is quite rare in oxaliplatin, but quite present when you treat patients with cisplatin. Even more worrying is this peripheral neuropathy. It's really a pungent, persistent pain at the extremity. And interestingly enough, it can last many years after the treatment has been completed. But while, for example, oxaliplatin gives an acute effect almost during the infusion or immediately after, in case of cisplatin instead, it can start paradoxically after the conclusion, the determination of the treatment. And therefore, you can imagine that from the clinician point of view, it's very difficult to anticipate and predict who will develop peripheral neuropathy and when, especially. So our group and other groups, of course, in the world have decided to convert, for example, this is the structure of a cisplatin. It's a platinum-2 species, extremely reactive, into a sort of prodrug that is a platinum-4 species that is basically including 2 groups here at the axial position. And the beauty is that this complex actually is quite inert. But I prefer saying that it's strategically active, meaning to say that it doesn't work when it is outside the cell, but when it is internalized inside the cell, it undergoes a reduction through reducing species such as glutathione, and then reconstitute the original, for example, cisplatin or oxaliplatin or carboplatin, according to what you start with, together with 2 additional functionalities. Now, the advantage of having these groups in red and blue is that not only in general the permeability through the cells is better, is improved in this platinum-4 compared to platinum-2, but most importantly is that these 2 additional groups enable in the introduction of additional functionalities. So, for example, you can introduce a targeting agent, a fluorophore, or another chemotherapeutic agent. And this is exactly what we did in collaboration with Professor Gibson from the Hebrew University of Jerusalem. And this is just an example of what we did. On one side, we introduced this 3-phenylphosphonio TPP molecule on one position. And on the other, we introduced, for example, some chemosensitizers, some dichloroacetate or PHB. Now, why we use the TPP to target the mitochondria is because, if you recall, I mentioned that the platinum-2 species have a biological target that is the nuclear DNA. But interestingly, also mitochondria are intracellular organelles that also contain DNA. However, differently from the nuclear DNA, the repair mechanisms within the mitochondria are much more defective. For example, the nuclear excision repair mechanism is basically absent in the mitochondria, and therefore we reason that if that is the case, then we can have an effect that is a point of no return for cancer cell. And indeed, we demonstrated that this occurred, the apoptosis is induced through a p53-dependent, caspase-3 mediated type of apoptosis. I would like to bring your attention to these two graphs. The first one indicates the accumulation of the platinum drug in the nuclei, and the other one at the mitochondria. Now, in the case of the accumulation at the nuclei, if you consider the references, the cisplatin and oxaliplatin, our best performer, the 3,4-cis, 3,4-oxa, as you can see, they are not that dissimilar in terms of amount that goes in the nucleus. But what is really remarkable different is the amount at the mitochondria. What does that tell us is that it's not that we are abrogating with our drug the effect at the nucleus, but rather we are adding an additional effect through the mitochondria. So what is our proposed mechanism? That you have a platinum-4, this is a just example, that permeates through the cell. Outside the cell does not work. Inside the cell is reduced. It forms a platinum-2. This is not in scale, because the nucleus should be much bigger, but this then goes to the nucleus with which it forms inter and intra-strands. But because there is also this TPP molecule, at least some of it goes to the mitochondria. And now what happens in case of cancer cell, they tend to become resistant to the treatment by making the membrane of the mitochondria impermeable to the release of this apoptotic inducing factor that would start the apoptosis. But by releasing the chemosensitizers, such as this dichloroacetate is a pyruvate dehydrogenase kinase inhibitor, basically what you do, you resensitize the cancer cell mitochondria towards the release of this apoptotic inducing factor that are here indicated in red, that we can see only when we have the administration of platinum-4. Now, I mentioned to you the side effect, the chemotherapy-induced peripheral neuropathy. And one recent experiment that we did is that we basically induced the peripheral neuropathy by treating animals with neurotoxic drugs such as cisplatin or paclitaxel. And then we measure different output, let's say. The first one is the mechanical sensitivity. So how much time does the animal take to retract the paw because it is too painful? And this is the mechanical sensitivity. Of course, you can see when you have a neurotoxic drug like cisplatin, paclitaxel, there is an almost immediate retraction of the paw. But when you have the cisplatin that is in a form of platinum-4, and here I would like to highlight the fact that the platinum-4 does not have additional targeting or functionalities. We simply have two acetyl group that don't do anything from the pharmacological point of view. But you see that the effect, basically the sensitivity, is decreased, so the animal experience less pain. And this type of effect, I don't want to enter too much in detail, but we confirm also when the animal is exposed to heat, cold, and even what we found out, these are just representative picture, but we have many, many more. We saw that when the animals, like little mice, are in pain, like with a neurotoxic drug, the eyes tend to be closed. So you see with cisplatin or Taxol, or even the physical mixture of the two drugs. So the eyes, in the majority of the cases, are closed. However, when you have a platinum 4, either cisplatin, the platinum 4 variant, or the combination of two neurotoxic drugs in a form of platinum 4, basically the majority of the time we could see animals with the open eyes. And the reason for that is that there is a net difference in the amount of platinum that accumulates in these DRG neurons, peripheral neurons that are then responsible eventually for the sensitivity that otherwise you see with the neurotoxic drug. So this from the, if you want, medicinal chemistry point of view. What about the delivery? Now, in case of delivery, you usually nanomedicines, the combination of a nanoparticle, a carrier, that incorporates and protects, usually in the inner core or on the surface, an active molecule. And you can use different materials. As you can see, you have different shapes, different sizes. You have the famous carbon nanotubes here. And we ourselves, we use a lot, for example, liposomes that are a lipid bilayer nanoparticles with an echo score. And this type of formulation is even approved by FDA, for example, in the case of Doxil for the treatment of cancer. Now, with this nanoparticle, irrespective of which material you use, they are usually in the nano scale range. When they are injected, they travel in the bloodstream. And when they reach the disease area, a phenomenon occurs. Now, I apologize because my schematic is is a simplification of what happens. But imagine that this one is the bloodstream, right? If you have a small drug like a platinum 2 or even a platinum 4, this drug is so small that can extravasate in normal tissue as well as in correspondence of the tumor site. And in the moment in which it accumulates here, you can understand, and then there are a lot of side effects. In the case of nanoparticles instead, they tend to be too big to extravasate in the normal tissue, and they tend to accumulate instead where there are this kind of fenestration or defective vasculature. And this phenomenon is called enhanced permeability and retention effect, because thanks to the fact that at the tumor site you have enhanced leaky vasculature, and to the fact that you have also defective lymphatic drainage, basically your nanoparticle tend to accumulate there, and therefore you increase a lot your bioavailability, the availability of your drug at the disease area. Now, which nanoparticle? I mentioned that we have been using quite a lot of drugs, but of nanoparticles, sorry. But recently we have conceived this kind of hybrid system. And this is made up of a lipid similar to the liposomes that I mentioned before, lipid-based nanoparticles. But on the other side, we also fuse it with a cellular membrane. Now, it is important to have this cellular membrane because on the surface, there are key proteins, lipids that improve the biocompatibility and even impart some targeting ability. In fact, in the case of liposomes that are even those that are used clinically, especially upon repeated administration, as it is in the case of chemotherapeutic agents, well, they undergo an accelerated blood clearance. That means that the body learns over time to recognize those synthetic nanoparticles, take them from the bloodstream and remove it. By instead having this hybrid system, you basically keep your nanoparticle not perceived as a non-self particle. And in terms of targeting, and this is just a small proof of concept, is almost my last slide, but in terms of targeting, I want to show you how we prove that there is an added value. At the tumor site, either endothelial cell or tumor cells themselves, they have a lot of these cell adhesion molecules on the surface. And we reason that if our hybrid system preserves some proteins that are able to recognize this cell adhesion molecule, when the nanoparticle reaches this area here, there should be enhanced uptake inside the target cell. So to prove this concept, we expose here endothelial cells in normal conditions. So there are some cell adhesion molecule, but then we expose them to TNF-alpha that tends to overexpress this cell adhesion molecule. And then we look, is there any difference in the uptake? And of course, as you know, the liposome, the lipid nanoparticle, they don't have any proteins, so there is not much of a difference between these two scenarios, and in fact, the uptake was the same. But in case of our hybrid system, we call it nanocell vesicle technology, you can see, first of all, the uptake is higher at the beginning because there is a recognition with a few cell adhesion molecule. But then in the moment in which you have many of them, you can see that the cellular uptake is almost double. And when you incubate the system with an antibody cocktail that blocks this cell adhesion molecule, you see that the uptake signal goes back to the baseline, indicating that these proteins not only are important and they are preserved structurally even upon the fusion, but also they help in the uptake. And we saw, in fact, that our hybrid system, in terms of uptake, there is a dose-dependent type of uptake, but it's much higher compared to lipid-based only nanoparticles. And this is also confirmed in vivo when you have that. Compare, for example, to the control. This is a colorectal cancer model where we consider how the tumor grows over time. Now, in case of a normal chlorocetate, so only the chemosensitizer or only PBS or the buffer, of course, the tumor grows over time. Cisplatin, because it's a potent molecule, as you can see, we administer 3 times over 3 weeks, and it works. But our platinum 4, even at half dose of platinum, worked equally well, and then a full dose of platinum was much more potent. But even more so when we loaded it into a nanoparticle. You can see the tumor basically was not palpable after 1 week of administration. So with this, I hope I have given you a little bit of an overview of what we do at the National University of Singapore. These are our 2 mascots, Mr. Pharmacist and Miss Pharmaceutical Scientist. And of course, I need to thank our very international bio-nanotechnology group, and of course, all of you for your kind attention. Thank you.
Thank you very much. This great talk and nice overview of your work. I think now it's time to justify why we award you, and there is a dedication that I want to say that you will find also on your certificate. This award recognizes not only Professor Pastorini's outstanding scientific achievements, but also her exemplary role as a mentor and inspiration to young researchers. She embodies the values of excellence, resilience, and curiosity that ESMEC strives to install to its alumni. Her success beyond national borders, driven by dissemination and brilliance, makes her a true model for the international scientific community and a distinguished ambassador for Italy, Europe, and ESMEC on the global stage.
Thank you very much.
I don't know if we have the certificate to show her, probably not, but you will receive it. We will send to you.
Thank you.
And thank you again, and congratulations for your work.
Thank you.
Okay, now I think it's time to move for the meeting of the young section of division of